Well, good morning, everyone. I am Elisa, and then today I'm going to talk about the prokaryotic diversity in the Southern Ocean in two contrasting productivity regimes. And first of all, the Southern Ocean has been characterized as the largest area with high nutrients with low chlorophyll in the global ocean. That means that despite the availability of nutrients, the primary production is not so high, and this is mainly attributed to the limitation by iron. However, in the surrounding of the Kerguelen Islands, there is an area that is mainly naturally fertilized by iron, and this sustains large phytoplankton blooms with the main peak during the spring, Austral spring. And then in this context of the iron limitation and the sites that are fertilized by iron with these with these phytoplankton blooms, the phytoplankton blooms can also act as a selective pressure over the prokaryotic communities. Actually, in some of the studies in this area has been shown that the heterotrophic prokaryotes are more abundant in the areas that are fertilized by iron than the ones that are low chlorophyll, in which the bloom doesn't occur. And even for the prokaryotic production, this can be more than twice higher in the areas in which the bloom occurs. So we know that our prokaryotes are responding different maybe in these two contrasting conditions. But what happened in the case of in terms of diversity? So this takes me to my research question, that is to know if the prokaryotic communities were different in terms of community composition and diversity in contrasting productivity regimes. And with that, my objective was to describe these communities in terms of composition and prokaryotic diversity over the Kerguelen Plateau, that is the area fertilized by iron, and outside of the Kerguelen Plateau. Also to determine some members of the community that can contribute to this difference if they are. So to achieve this, it was an oceanographic cruise took place during February and March of 2018. And there were 3 sites visited. The site M2, that is over the plateau. The plateau is here delimited by the bathymetry. And then that is characterized to having a high biomass. And the other stations that are outside of the plateau that have low biomass. In each one of the stations, seawater was collected in 4 depths, 10, 60, 125, and 300 meters depth. And then the water was filtered to obtain 2 size fractions. The prokaryotic communities that are filtered in a sieve of bigger than 0.8 micrometers, and this can be attached to particles. These particles can be either organic matter, detritus, for example, but also they could be maybe alive cells. And we also retrieved the free living fraction, that are the prokaryotes that pass through the sieve, smaller than 0.8 micrometers. Then it was performed the DNA extraction for analyzing the total community composition, and also the RNA extraction because it's used as an indicator of the active community. Then it was sequenced the 16S gene via Illumina. And when the sequences retrieved, they were mainly trained in the software R with the package DD2. First, I selected the ones that were with good quality. And also, it was made the inference of the amplicon sequence variants. The amplicon sequence variants are groups of sequences that are as different to other sequences as smaller as 1 nucleotide. In this case, they are going to be your taxonomic units to have this term. So ASVs or taxonomic units, for the ones that are not familiar with these terms. It was calculated the abundance of these taxa in the samples, and the taxonomy was assigned using the PhyloSec package and against the Silva database. Also, they were calculating the diversity indices, and this information was used for the statistical analysis. To answer the questions, if the communities are different, it was performed a Brian-Curtis dissimilarity. that also was used to build an MDS ordination. Then to test for significance, it was perform a nanosim analysis. And to know which taxa are contributing to these differences, it was performed a simple analysis. All this was made with a vegan package for R. Then let's see how were the conditions during this sampling cruise. All the stations visited were stratified in the water column, and the surface mix layer was around 60 meters depth. But we can see here that on the plateau side is slightly higher the chlorophyll values than in the case of plateau. And however, the maximum is around 0.6 milligrams per cubic meter. That corresponds to post-bloom conditions. So it's not the maximum of the peak of the bloom. It's after that. So now, to see how our prokaryotic communities are distributed in the area, I'm going to use this kind of ordination that is an NMDS, non-metric multidimensional scale. And for the ones that are not familiar, this is an ordination that allows you to see the differences between the samples in 2 dimensions. And then here, each one of the dots corresponds to one sample, and the dissimilarity was calculated based on the community structure, either if the ASBs were present, absent, or also the relative abundance of each one. So here, we can see, if we take all our samples, that the prokaryotic communities were clustered by the depth layers, being the 300 meters communities different from the ones in 125 and different from the ones that were in the surface mix layer. This is for the free-living communities. In the case of the particle-attached prokaryotes, we found the same clusters. However, the pattern is slightly less marked. And this, well, you can ask maybe why this is more marked in the case of the free-living prokaryotes. The stratification of the water column can act as a physical barrier for the free-living prokaryotes, so they are well separated depending on the water depth. And then, in the case of the particle-attached ones, as they are unique to some particles, these particles can easily sink in the water column, providing more connectivity among the water layers. Now, if we focus only in the surface, that is 10 meters depth and 60 meters depth, we saw that the free-living communities, the samples that were over the plateau are different from the ones that were outside of the plateau. And this is for the free-living and the particle-attached ones. Also, well, they are different in terms of, for example, diversity. Here is an example of one of the indices of alpha diversity that was calculated. And you can see that over the plateau is lower the diversity in comparison to outside of the plateau. How we expect this, how we can explain this, maybe for the predominance of a few taxa that are dominating the community due to the conditions that are present there. They can be fast growers, for example, or opportunistic ones. In the case of the particle-attached fraction, we can, an important or interesting pattern to notice is that in comparison to the free-living ones, they are less diverse. And then this can maybe be related to the particle types and actually with the specificity that the certain taxa can have for certain particles. Now, we see that the sites are different, and to know which taxa are contributing to this difference, I'm going to show you some numbers here. I take as an example one of the comparisons I made. If we take only one side outside of the plateau and one side over the plateau, we retrieve around 5,000 taxa. From them, according to the simple analysis, only 93 taxa were contributing significantly with the differences between groups. And from them, I then selected manually the ones that were more than 5% relatively abundant in at least one sample, that is highly abundant in this case of the communities, and they were explaining almost the 40% of the difference. So I took these 11 taxa for the next figure. This is a heat map in which you can see with the color code the relative abundance, being the darkest, the most abundant ones. And the difference between on the plateau and outside of the plateau for the total community and for the active community. Here, this figure is only for the free living fraction. However, also perform the analysis for the particle attached, and then there are some taxa that are actually common in both. And as I cannot talk about all of them, I will take two examples that are highly relative abundant over the plateau, and two taxa that are higher relative abundant in the outside of the plateau. So let's take a look of what kind of things these guys do. In the case of the site that is over the plateau, enriched by iron, and in which the phytoplankton blooms occur, there is this licase that, according to the literature, has been observed occurring with algal blooms. That, well, makes sense. This is one area that is also predominating with algal blooms, so okay. And then also via some transcriptomic analysis recently, it has been shown that they can, some groups related can express genes related to the iron uptake that can give them an advantage in terms of the iron enrichment in this area. Maybe against other members of the community. Then we have Flavobacteriales, and this NS5 marine group that has been shown with a high correlation with other, with some group of diatoms that are actually important in the Kerguelen blooms. But also this Flavobacteriales has the capability of degrade fast the polymers. Now, if we move to the ones that are outside of the plateau, we can see that the Flavobacteriales again, but this is a different genus, Formosa, and this can degrade also polysaccharides rapidly and effectively. And okay, both are the same family, but then they are different genus, and well, both has this fast polymer degradation. Maybe the difference is about the specificity for the organic matter that they prefer. And then we have SAR11 that is also highly abundant. It's one of the most abundant bacteria in the global oceans. And it has been observed as a good competitor in all of eutrophic environments. So it makes sense that it's in this site outside of the plateau. To sum up, then the depth was a constraining factor for the prokaryotic communities in the Kerguelen region. And in the surface water, we observed the differences between the communities in both regimes. Also in terms of composition and diversity, being less diverse, the communities on the plateau in comparison to the ones that were outside of the plateau. And if we, according to the simple analysis, the 40% of the difference was explained for 11 highly abundant taxa that can be due maybe for the adaptive capabilities they have. Now, we need some other things to understand. For example, what is the nature of these particles to which our communities are attached, in the case of the particle attached communities, of course, and maybe they are having some effect on them. And if the difference in the prokaryotic communities is also related to the phytoplankton assemblages in the area. And also would be good to know, and interesting to know, the functional properties of these most competitive taxa that further than the literature can be done by transcriptomics or proteomics approach. And with this, I would like to thank you all for your attention. And yeah, myself.